So my laboratory is in Riyadh at King Faisal Specialist Hospital and Research Center, and we are interested in understanding basic mechanisms regulating cardiac hypertrophy and heart failure. We look at epigenetic mechanisms, also genetic causes in the Saudi population, but today I thought I would present you some result, a study that we have published recently on the genetic ablation of a gene, which is called ACP1, which encodes a protein, Lmptp. So for you to remember, ACP1 is equal to Lmpt1 to make it simple. And so we have deleted this gene in mice, and the mice, I will present the data, reveal the pretty striking phenotype. Which one is it? Okay. This one. Okay. So as all of you know, cardiovascular diseases are really a burden to our society. They are the leading cause of morbidity and mortality worldwide. These are the numbers from the World Health Organization. And so in Saudi Arabia, cardiovascular disease account for 25% of all death. With a population that is truly young, 80% under age 40, the incidence of death from cardiovascular disorders will rise. And it is estimated that it will cost 80 billion Saudi riyal to the society in 2030. These are examples of two hearts that we collected from patients that have undergone cardiac transplantation with the help of our colleague clinicians at the Heart Center at King Faisal Hospital. The one on the left is dilated, shows clear dilation of the left ventricle with a lot of fibrosis. The one on the right is hypertrophic with reduction of the ventricular chamber. So as a basic scientist, what the questions I ask is what are the mechanisms that lead to those two pathologies? And you as clinicians are asking, when is it that you're going to find a cure? And as of today, there is no cure for heart failure, and clinicians manage heart failure, and scientists are very frustrated into trying to understand, how to discover new treatments. And one of the reasons why it is so difficult is, I think, illustrated pretty well in this slide. These are just a summary of the major signaling pathways that are changed in cardiac hypertrophy. And the point that I want to make, and for you to remember, is that most of the basic studies have investigated roles of protein kinases, and very few have focused the roles on protein phosphatases that act in opposite manner to protein kinases. There are 3 studies that have been published that have been done in mice showing that deletion of the phosphatase SHP2, it's a cardiac-specific deletion that causes dilated cardiomyopathy in mice. Another one where protein tyrosine phosphatase 1b was deleted was actually protective. And this one where inactivation of PTEN by gene deletion or pharmacol inhibition also protected against ischemia reperfusion. So we are interested in another protein, tyrosine phosphatase, which is called Lmptp. What is it? What is this protein? It's actually the smallest of the protein tyrosine phosphatase. It belongs to its own class. It's expressed everywhere, and it's encoded by a single gene called ACP1. Here's the gene structure, and there are alternative splicing give rise to 3 transcripts that are called IF1 and IF2, which encode 2 different proteins with a very different biochemistry. The 3rd transcript is actually non-coding. So what was known about this protein when we initiated our studies is that basically there were some indication that this protein may be a key regulator of metabolic homeostasis. There were some studies in vitro biochemical studies showing that the phosphatase regulates insulin signaling by dephosphorylating tyrosine on the insulin receptor. This gene, the gene itself, is highly polymorphic in the Western population, and polymorphism affects the enzymatic activity in human. And in general, low activity of the phosphatase is associated with protection against metabolic syndrome in human. And there was one study in mouse that had shown that obese mice, when treated with Lmptp antisense nucleotide, have improved insulin sensitivity and reduced hyperglycemia. But what is the role of the phosphatase in the heart? We have initiated studies by looking at the expression of the protein during early development. And you can see on the left panel here that the phosphatase is expressed very highly in the embryonic heart and then declines after birth, although the levels still remain detectable. And what we have seen in mice is that when you subject the mice to pressure overload hypertrophy, you have increased expression of the phosphatase in response to stress. And we have performed also a high-resolution echocardiography here to show you that those mice indeed were responding to stress. How about in human failing hearts? Since we are collecting a lot of idiopathic dilated cardiomyopathic hearts, we also assess the expression of the phosphatase both in control in the heart of normal individuals and in the hearts of patients with idiopathic dilated cardiomyopathy. And you can see there's a striking increase in the expression of the protein. So suggesting that pathological stress is associated with its upregulation. And another thing that we also verified is that those failing hearts, and I just showing you two panels here, they have increased fibrosis as opposed to control hearts. So we decided to delete the gene in the mouse, and we did that by inserting a gene in the genome after the fourth exon, and this is to show you that indeed the wild-type mice express the protein, the heterozygous mice expressed half of the amount as expected, and the knockout mice expressed no protein at all. So our targeting strategy was successful. We have performed a lot of, we have made a lot of effort in trying to survey all the tissue, and first we analyzed the mice at baseline with no stress. And what we saw is that the mice in all the organs that we have surveyed showed no signs of pathology at baseline. They develop normally. At 10 weeks, they are a little bit smaller than the control mice, but then it looks like they catch up. So this is a mouse that's very happy, that has a long, normal lifespan, nothing special. So the next thing that we did is to stress the heart by creating a ligation of the aorta to create pressure overload. And then we performed high-resolution echocardiography every week for 10 weeks. At the end of those 10 weeks, we sacrificed the mice, collected the heart, and then performed biochemical analysis. So here I'm going to give you a summary of the echocardiography, and I think that you can appreciate as clinicians that the knockout mice, and you have to look here at the curve in red compared to the blue one. So while the wild-type mice develop hypertrophy at 5 weeks post-TAC and then go into failure, and this is indicated here by the increase in ejection fraction initially, and then it goes into the decompensated phase, and the function is severely compromised. Here you're looking at the end-systolic volume. We have more parameters, but this is just to summarize. So the knockout mice are protected against this deterioration, both of the ejection fraction. The wall thickness remains mostly normal, septum thickness, the same thing. So we were pretty excited about that. And at study endpoint, if you just look at the size of the heart, you can see that the wild-type heart is quite enlarged, while the heart of the knockout mice is not as enlarged. It's pretty obvious and it's significant. If you are performing staining to look at the size of the cells in the tissue of these mice, you can also appreciate that the wild-type mice have enlarged cells, and the knockout mice, after stress, again, resemble more of a sham-operated animal. We also have data showing that fibrosis is severely reduced in these animals. This is just a slide to illustrate the extent of fibrosis by trachrome staining. In the sham-operated animal, they are pretty normal. And here, this blue staining is very prevalent in the wild-type animals and not as much in the knockout mice after TAC. And we have done a quantitation of this. In addition, we surveyed heart failure markers, and these are also not as strongly upregulated in the knockout mice subjected to TAC. So we are interested in, we have this exciting phenotype, and for us is what is the mechanism behind this? So to try to answer this question, we performed gene expression profiling in all the 4 groups of animals. And this is just a heat map representing all the genes that are dysregulated in the sham-operated animals that are wild type or the knockout mice. After pressure overload, you see that the pattern is changing significantly in the wild-type mice, and the knockout mice subjected to stress also have a very different pattern. And here are the number of genes that are globally up or down regulated in the wild-type mice, and you can see that genes that are actually, when you look in details into the list of those genes that are upregulated in the wild-type mice after pressure overload, they are actually linked to hypertrophy, which makes a lot of sense. So we looked into the details of those genes, and what we found out is that in general, deleterious signaling pathways are actually minimally upregulated in the knockout mice that are subjected to stress. And this is indicated here by, if you compare this lane versus this lane, for instance, the phosphorylated 4-CAMK enzyme is not as strongly upregulated, and so are those proteins, PLC beta, and so is the G-alpha-Q receptor. This is just an example. And on the contrary, we found that protective signaling pathways are actually strongly upregulated in those knockout mice after stress. This was the case of the insulin receptor and some other pathways here that we are now investigating in more details. And here is just a Western blot showing you the type of difference that you can expect. This is the level of phosphorylation of the insulin receptor in the mice that do not develop heart failure in comparison to the other mice here. So, in summary, what we have found, and we are very excited about that, is that deletion of this gene protects against cardiac hypertrophy and heart failure in mice. We have one mechanism which involves minimal activation of protein implicated that are deleterious in general, and hyperactivation of signaling pathways that are known to play a protective role in the heart, such as phosphorylation in the insulin receptor, but there are other pathways as well, which we are studying now. Now that we have this, what do? We want to go deeper into the mechanism, and that's what we are doing by generating conditional deletion of the phosphatase in cardiomyocytes, but also, in fibroblasts in relationship to the talk of the previous speaker. We are also screening for small molecules inhibitors because we have the tools to do that, and testing them in mice to see if they can prevent heart failure. And the hope is that we will be able to move to translational research if we are successful in having a good candidate. This is to acknowledge the people that did the work in my lab, and also some international collaborators and local collaborators, Dr. Walid Al-Habib, with whom I have, I can say now, a long-standing relationship. Thank you very much.
Thank you.